In recent decades, events have been unfolding behind the closed doors of research centers that once seemed like the stuff of science fiction. In the early 2010s, sensational reports began to appear around the world about people who suddenly got the chance to become younger not just in appearance, but biologically as well. In one of the laboratories at the University of California, San Francisco, scientists used complex analysis to show that the biological age of some experiment participants decreased by several years in just a few months. The foundation of this was a protocol that included a unique combination of growth hormones, metformin, and vitamin D. One of the researchers, Dr. Gregory Fahy, even participated in the experiment himself. According to the analysis of epigenetic markers, his biological age decreased by 2.5 years, even though only one actual calendar year had passed. This case sparked a strong reaction in the international scientific community. Meanwhile, in Israel, at the Weizmann Institute, similar experiments were conducted on people over 70 years old. After the course of treatment, their biological age decreased by more than a year compared to their actual age, and their immune cells began to behave like those of much younger people. European scientists from Germany conducted their own analysis and found that after bone marrow transplants from young donors, elderly patients began to recover much faster and even reported feeling subjectively rejuvenated. But an even greater interest was sparked by a series of experiments at Harvard when Professor David Sinclair proved that it is possible to activate so-called youth genes by simply influencing DNA with certain compounds. These discoveries became the starting point for fantastic research that is now ongoing all over the world. However, behind each case, there is not only complex biology, but also an amazing set of factors that happen to coincide at a particular moment in time. In the next stories, we will find out whether hormones can really grant a long life. Or is it just a myth that arose from the first seemingly miraculous cases? Longevity hormones, reality or myth? When it comes to hormones that can prolong youth, science inevitably encounters astonishing stories of sudden rejuvenation and incredible results. At the end of the 19th century, French physiologist Charles Edouard Brown-Séquard was the first to inject himself with an extract from the testicles of young animals. According to contemporaries, although this experiment did not produce a lasting effect, it caused such a stir in scientific circles that the idea of hormonal rejuvenation remained in the minds of scientists for many decades to come. In the mid-20th century, the first experiments with growth hormone began at major universities in the United States. Volunteers who received injections over several months reported a surge of energy and even an increase in muscle mass. In recent times, scientists from Stanford have observed people whose natural levels of somatotropin remained at youthful levels. They truly maintained physical activity and robust health longer than their peers. But the price of such a condition often included risks to the heart and a tendency toward tumors. At the same time, experiments were being conducted in Copenhagen with the so-called youth hormone melatonin. Researchers gave it to volunteers over the age of 65 and recorded improved sleep quality, faster recovery after exertion, and even increased bone density. However, with prolonged use of hormones, some participants began to experience metabolic disorders. Therefore, after a series of studies, their use was limited to only the most severe cases. On another continent, in Japan, an experiment was conducted with the hormone DHEA, which is produced by the adrenal gland. People who participated in the program reported reduced fatigue, feeling energetic in the mornings, and even the disappearance of wrinkles. Journalists have repeatedly raised the question of whether it is truly possible to prolong youth with this compound. The answers remained ambiguous, as scientists continue to propose new hypotheses about side effects. At Harvard Medical School, special attention was given to a hormone called IGF-1. Studies on mice have shown that lowering its levels does indeed extend lifespan, but also increases vulnerability to certain diseases. Despite impressive reports and stories from experiment participants, medical organizations remain extremely cautious about hormonal interventions to this day. One of the main reasons is the wide range of results, from noticeable rejuvenation to dangerous side effects. While some scientists in Boston are studying hormone combinations to create real elixirs of youth, others in Switzerland are trying to accurately predict the body's reactions using supercomputers. But all these studies have gradually led to an even more mysterious discovery, which is not related to injections or pills. In the next part of the story, we will see how the secrets of longevity are hidden not only in hormones, but are literally encoded in the very DNA chain itself. A discovery in DNA. Restarting the aging clock? Delving deeper, scientists began to study DNA from a new perspective and not just as an immutable code, but as a complex mechanism that, if not hackable, can at least be reprogrammed. It was precisely such experiments that caused the sensation in Massachusetts laboratories when Professor David Sinclair first proved that chromosomes age not only due to chronic damage, but also because of the switching on and off of so-called clock genes. In one of Sinclair's group's experiments at Harvard, laboratory mice had their aging artificially accelerated through chemical exposure and then, using a cocktail of 3 factors known as the Yamanaka factors, their biological clocks were literally reset. The result was so striking that the old mice restored the function of their organs, their fur became thick again, and their vision returned to its previous level. DNA analysis showed that the age-related epigenetic marks in the mice had almost completely disappeared. Following this, a startup in Palo Alto led by Dr. Jose Cozo attempted to replicate the experiment on monkeys. The use of individual factors made it possible to slow down the aging process of skin and heart tissue cells. At the same time, a laboratory in Cambridge announced the development of the reset method for human cells in vitro. In a test tube, old fibroblasts taken from patients over 80 years old, after a brief treatment with a special mixture, began to divide and function just like those from young donors. At the same time, a team from Munich presented sensational data. It turns out that long livers from the Caucasus living in the mountains have a unique epigenetic profile. According to sequencing results in these people, the very DNA elements responsible for immunity and the body's regeneration remain active much longer. This discovery gave new impetus to the development of epigenetic age tests, which are now available in several major medical centers in the United States. But one of the most mysterious discoveries was the experimental CRISPR therapy, which began in the 2020s in a Texas laboratory. For the first time, doctors attempted to modify specific sections of DNA in patients with rare aging-related diseases. As a result, some test patients showed a slowdown in the progression of symptoms and improvements in skin and vascular condition. In laboratory-grown tissues, scientists observed an almost complete restoration of structure. Researchers continue to search for new approaches to target the so-called longevity genes. Already today, startups are emerging that offer epigenetic correction based on individual profiles. And in Singapore, investors are pouring millions of dollars into cell reprogramming technology. If in the past the secret of eternal youth seemed like something out of fairy tales, today everything points to the fact that the key to this mystery already exists within the very structure of DNA. But nature itself has long created astonishing mechanisms of longevity in animals, and the next discovery can be considered almost fantastic. Ageless animals, nature's secrets now accessible to humans. When scientists encountered the mystery of biological youth, their attention was immediately drawn to creatures whose bodies seemed to be untouched by age. One of the most astonishing discoveries took place in a laboratory in Woods Hole, Massachusetts, where researchers were studying the so-called immortal jellyfish Turritopsis dornii. These tiny creatures literally reset their life cycle. Instead of dying, they reverted to the polyp stage and began developing all over again. This phenomenon was first recorded at the end of the 19th century, but it was only in the early 2000s that biologists conducted a detailed analysis of the jellyfish's cells and discovered unique proteins that triggered the rejuvenation process. At the same time, biologists at New York University were studying long-lived bowhead whales. These Arctic giants regularly live to be 200 years old and maintain healthy blood vessels for much longer. Sequencing their DNA revealed several mutations that block cellular aging and prevent the development of malignant tumors. Scientists have identified specific regions of the genome responsible for rapid tissue regeneration and are now developing models to simulate these mechanisms in humans. The so-called naked mole rats from East Africa have also attracted enormous interest. These colonies of burrowing rodents live in conditions where the average lifespan of other animals does not exceed a few years. But mole rats easily live to 30 years or more, showing virtually no signs of aging either externally or internally. A group of biologists from Cambridge found that their cells undergo almost no age-related changes and their immune system remains active even in old age. Studies have shown that the proteins in their bodies are able to quickly repair damaged sections of DNA, a discovery that is already being used in experiments to extend the lifespan of laboratory mice. In Texas, a series of experiments was conducted with albino fish living in the depths of the ocean, whose metabolism is so slow that age-related changes in their tissues are almost imperceptible. Scientists have observed that such fish are capable of repairing damaged cells even after decades of life. Currently, biochemists are testing similar proteins on human cells in vitro to find out whether it is possible to reprogram the cellular cycle. But the most mysterious of all turned out to be planarian worms, which zoologists have known about since the early 20th century. In a recent experiment at a laboratory in Illinois, biologists split an adult specimen into two parts, and both halves regenerated the missing organs without showing any signs of aging. Geneticists have already identified the genes responsible for this process and are testing the possibility of activating them in mammals. All these examples show that regeneration, immortality, and control over biological age already exist in nature. Now, scientists around the world are working to incorporate at least some of these mechanisms into medical biotechnology. Engineering experiments are underway to insert longevity genes into human cell cultures, longevity proteins are being modeled, and drugs that affect DNA repair are being tested. But a real breakthrough may be closer than it seems, as some results have already shown fantastic effects in laboratory conditions. However, even nature, with its centuries of evolution, does not provide a ready-made recipe for eternal youth for humans. That is why scientists are turning to the technologies of the future and the boldest experiments in an attempt to turn back time. Laboratories of the future. Experiments to reverse aging. While natural wonders inspire biologists to search for new solutions, a real technological race is unfolding in modern laboratories, where each experiment challenges the boundaries of human life. In Oxford, UK, a team of doctors led by Andrew Steele has been testing a method based on blood plasma replacement for several years now. As part of the project, all the volunteers received transfusions of plasma from young donors. After a series of protocols, doctors observed a sharp decrease in inflammatory processes in the subjects, improved memory, and even accelerated muscle tissue recovery. Similar tests were launched simultaneously at Stanford, where scientists noted that after a course of transfusions, some aging biomarkers returned to levels typical of people 1 or 2 decades younger. No less impressive results are being demonstrated by bioengineers from Osaka University. In their laboratory, miniature organoids have been created in copies of human organs grown from volunteer cells. Hundreds of compounds capable of triggering cellular rejuvenation are being tested on these models. One series of experiments showed that exposure to certain molecules initiates the regeneration process, making old cells function as actively as young ones. At the same time, in Copenhagen, researchers are trying to use DNA editing with CRISPR technology on patient stem cells. Even the initial trials have yielded data on the restoration of vascular elasticity and improved heart function in laboratory rodents. Now, the first clinical tests for humans are being prepared. Special attention is being paid to projects focused on reprogramming cellular age. And the leader in this field is considered to be a company from Boston, where a team of scientists led by George Church is experimenting with cocktails of proteins, signaling molecules, and epigenetic modifiers. In the laboratory, they treat skin and liver cells which within a couple of days lose signs of aging and restore their structure and metabolism. Methods for rejuvenating bone marrow and neural tissues are already on the way. And in Israel, a clinic affiliated with the Technion is beginning trials on patients showing signs of early aging. After a short cycle of procedures, some volunteers have shown improved immune response and accelerated wound healing. In laboratories in Southern California, microparticles are simultaneously being developed that can deliver rejuvenating substances directly to target cells, bypassing side effects. These nanocontainers are currently being tested on old mice, and according to the results of the first series of experiments, their liver and heart are showing characteristics typical of young individuals. Meanwhile, in Zurich, an automated artificial intelligence system is operating, analyzing hundreds of thousands of molecules and predicting their potential to combat aging. As a result, within just a few months, several new compounds have been discovered in the laboratory, and they are now undergoing preclinical testing. All these experiments show that in the laboratories of the future, it is repeatedly possible to slow down or even reverse the aging processes at the level of cells and tissues. But the main mystery why the seemingly impossible sometimes works remains unsolved. True breakthroughs often happen where conventional methods fail, and it is often chance that becomes the starting point for the most unexpected discoveries, as evidenced by a whole series of historical examples that are still waiting for their moment. Immortality technologies, nanoparticles, and artificial intelligence. Cutting-edge laboratories in the US and Europe are putting into practice technologies that, until recently, seemed unbelievable even to seasoned experts. At a laboratory at the Massachusetts Institute of Technology, nanoparticles for targeted drug delivery were created as early as the beginning of the 21st century. Microscopic containers less than 1,000th of an inch are loaded with drugs that activate regenerative processes in tissues. In one experiment conducted in Boston, aging mice were injected with nanoparticles containing a growth factor. After a few weeks, their condition resembled that of young animals. Bone density, vascular elasticity, and even wound healing speed had nearly doubled. During the same period at the University of California, a group of bioengineers, led by Jennifer Doudna, conducted a series of tests with nanoparticles capable of reading aging signals on the cell membrane and activating protective proteins only at the right moment. This made it possible to minimize side effects and increase the efficiency of active substance delivery several times over. Later, Japanese colleagues used similar structures to treat age-related changes in the retinal tissue of laboratory monkeys. Vision was fully restored within a few weeks. At the same time, artificial intelligence is becoming the main assistant in the search for new compounds. In London, a neural network has been created that analyzes millions of results from previous studies and generates hypotheses about potential elixirs of youth. A prototype of the system, implemented in Zurich, identified 35 new molecules with signs of blocking liver cell aging within 3 months. In San Francisco, experts use machine learning to predict how human DNA will react to different combinations of drugs, which speeds up obtaining results from 6 months to just a few weeks. At the University of Toronto, automated robots conduct hundreds of tests daily, mixing nanoparticles with biological agents and recording when a cell begins to repair damaged sections of DNA. One of the experiments led to the creation of smart nanoparticles that independently determine the extent of damage and activate the appropriate set of repair genes. The first wave of tests showed that skin cells from elderly volunteers, after being treated with these particles for 48 hours, began producing collagen at the level of 20-year-olds. In New York, The startup in silico medicine implemented artificial intelligence to discover new classes of antioxidants that increase the lifespan of laboratory rodents by nearly 30%. Data sets are processed around the clock, and now, every quarter, information about dozens of previously unknown molecules appears in international databases. But it is precisely the combination of nanotechnology and artificial intelligence that has become the platform for the first personalized anti-aging program. Several clinics in the United States are already using comprehensive genome analysis of patients, after which robots select a unique cocktail of nanoparticles and auxiliary molecules targeted at specific signs of aging in that individual. The first patients report restored muscle tone and improved memory after just 4 to 6 weeks of therapy. All of this is happening right before the eyes of modern researchers, and there is no doubt that such a technological leap became possible thanks to unexpected discoveries and accidental results in the most seemingly routine experiment. What remains is to find out what lies behind these moments of insight, when a breakthrough occurs not because of a plan but in spite of it. The unexpected truth. Why are discoveries always driven by chance? It is precisely chance that has repeatedly changed the course of experiments, leading to discoveries that have forever transformed the science of aging. Back in the late 19th century, the French doctor Louis Pasteur was searching for ways to create a vaccine when one day he noticed that a forgotten chicken culture in the laboratory had produced weakened bacteria that did not cause disease but instead formed immunity. This missed experiment became the starting point for modern immunology. Decades later in Cambridge, a young researcher named Alexander Fleming was conducting routine observations of bacterial cultures. By accidentally leaving the petri dishes uncovered, he noticed that the bacteria around the mold had died. This is how penicillin was discovered, and with it, the era of antibiotics began. In the 20th century, biochemist Paul Lauterbur was experimenting with radio waves and accidentally placed a magnet in the wrong position, which led to the development of the MRI method. Now it is impossible to imagine modern research on age-related changes in the brain and tissues without it. In California, Japanese professor Shinya Yamanaka was searching for a way to reverse cell development, and one day, just to test a side hypothesis, he added 4 genes to a cell culture. As a result, he managed to reset the cell's biological clock and obtain starter stem cells from ordinary tissues. This case became the foundation for modern cellular rejuvenation technologies. The story of Jennifer Doudna and Emmanuel Charpentier with the CRISPR technology also began with an unexpected discovery. While analyzing bacteria that survived viral attacks, they accidentally noticed strange sequences in the DNA, which later turned out to be a natural gene editing mechanism. Today, this process allows scientists to precisely intervene in the genome, slowing down aging. In Israel, during the development of smart nanoparticles for tumor therapy, biochemists noticed a side effect for the first time. In elderly mice, the nanoparticles activated not only the immune system but also genes associated with tissue regeneration. The final series of experiments showed that the old animals literally began to rejuvenate, even though the researchers were originally looking for a different effect. In Massachusetts, a project to automate molecular analysis using artificial intelligence was launched with the goal of speeding up the search for cancer drugs. But the initial results unexpectedly led to a group of compounds that accelerate tissue renewal in aging laboratory animal models. These compounds are now being used to develop new rejuvenation programs for humans. In the laboratory at New York University, scientists were testing automated drug mixing systems. One day, a software glitch caused one of the cell cultures to receive a random combination of 8 molecules. The next day, the cells showed rates of division and regeneration comparable to those of young samples, although previously such compounds had never been used together. In Cambridge, a research group working with long-lived volunteers accidentally discovered a rare mutation in one of the participants that grants immunity to age-related inflammation. This case became the basis for the creation of new aging biomarkers and tests that are now used by hundreds of medical laboratories. Thus, every modern breakthrough in rejuvenation and life extension often begins with a simple mistake, an unexpected result, or equipment failure. From a random mutation to an unexpected reaction of cells in a test tube, these very moments become the catalyst for true scientific revolution. And perhaps the next key to the mystery of eternal youth is not hidden in a precisely calculated laboratory formula, but rather in that very unexpected coincidence that will turn yet another experiment into a breakthrough for all of humanity.